So we're going to continue talking about decision making, and we're going to be investigating it at the level of detail of neuroscience. And so there's a foundational idea, obviously, that underlies the creation of these decision making concepts and models that we've been talking about. And this is the fact that before we can actually make a decision, we have to first come up with the value of each of the options. So first we have to judge how valuable our various options are, and then to engage in some sort of comparison between said values. And so this brings up the question about how and where is value represented in the brain. It also brings up an associated idea of, well, is decision making, is it all kind of located in just one place? Or are there various other areas that work together throughout the brain? And so I'm going to be talking about some more updated information in comparison to your textbook, just to continue to nuance this conversation and to push it farther with more updated information that we have. And so researcher John Wallace and his colleagues in 2009, they looked at the representation of value in the frontal lobes of monkeys. And they were particularly looking at the frontal lobe because we know that damage to the frontal lobes is associated with impairments in decision making and our goal-oriented behavior. And so while the monkeys were performing decision-making tasks, the details of which we will get into in just a second, these investigators were using multiple electrodes to record from neurons in 3 different areas in the monkey brain. So they were looking at what's called the anterior cingulate cortex, the lateral prefrontal cortex, and the orbital frontal cortex. And again, these are all areas much further forward in the brain. So in addition to comparing cellular activity in these 3 different locations, the experimenters also manipulated what they called the dimensions of cost, probability, and payoff. And so they were essentially altering these 3 sort of conditions in the decision-making paradigm that the monkeys were given. And the reason for this was because they were trying to answer the key question of whether the different brain areas would show selectivity to particular dimensions. That is, do the different areas provide support and neural activity in these different sort of aspects of decision making. So the monkey's decision involved choosing between 2 different kinds of pictures. And this is why there's a picture of a guitar and a flower on the screen. And each picture that was shown to the monkeys was associated with a specific value of one of the 3 dimensions of either cost, probability, or payoff. So, for example, an image of a guitar was shown to monkeys, and this image was always associated with the payoff of 3 drops of juice. And the picture of a flower was always associated with 2 drops of juice as the payoff. And so on any given trial, the monkeys' responses were being influenced by, like I said, variations in these dimensions. And on these trials, the choices that they could make, they were based on manipulations of one dimension while the other two dimensions stayed constant. So they weren't changed by the researchers. So for example, when the dimension of cost, let's say it's measured in the number of lever presses that the monkey had to make in order to get a reward. When cost was what varied in between the pictures, the probability and the payoff. So, for example, the specific amount of juice was constant. Um, and so, again, they're altering these different dimensions to see if there were differences within these sorts of these features of decision making, of cost, probability, and payoff. And also want to point out that each area of cells that was investigated across the 3 different regions involved cells that would respond to multiple dimensions of decision making. So it's not just that, they were just looking at cells that would respond to cost in the ACC, for example. But really trying to look at the interaction between these dimensions and regions. And interestingly enough, there wasn't a really clear distinction between the representations of these 3 dimensions. There were some notable differences, however. So first, a lot of the cells, especially the ones in the anterior cingulate cortex, but also at least a noticeable amount in the orbital frontal cortex as well, responded to all 3 dimensions. And so this kind of pattern suggests to us that the cells in these particular areas represent what can be considered as an overall measure of value. So the cells in the anterior cingulate cortex and some of the cells in the orbital frontal cortex were responding to information in a richer way. So it wasn't that they're just looking at one aspect of a dimension, such as the cost of a choice or the probability, but really taking in a broader range of information across those dimensions. But then in contrast, the cells that were in the lateral PFC, they usually just encoded or focused on just one dimension variable. So there's more specificity that was seen here in that region. Wallace and colleagues also found that for the neurons that responded to just one of the dimensions, it usually was either payoff or probability. So again, if a cell was being more specific and sensitive in its function, it was focusing more on the probability of the situation or the payoff, and not so focused on the potential cost that came with a decision. And then finally, the value signal in the cell's activity generally preceded the activity that was associated with motor preparation. And essentially, this suggests that choice value is being computed before the response is selected. Essentially, the value with which we are assigning something is done before we even begin to make a response, even if this is just small, maybe unconscious sorts of motor movements. And this is an observation that makes intuitive sense, right? That we're having to run all these different calculations before we can even begin to move our bodies to help execute in its entirety that decision. And so there have been similar studies done with human participants. And this has been done with fMRI scanning. Um, and so here, the emphasis has been on how these sort of different dimensions of decision making might activate certain neural regions. So, for example, in one study, orbital frontal activation was pretty closely tied to um, uh, the dimension of payoff. Whereas activation in the striatum of the basal ganglia, and we know that the basal ganglia is crucial to motor movements, the activation here was related to someone's effort in following through on the decision that they had mentally made. In another study, there was lateral prefrontal cortex association with the probability of a reward. And also, researchers had also looked at the delay between the time that an action was executed and then an associated payoff. So essentially, here we are looking at what kind of brain areas are involved with not only our initial execution of an action when we're working towards a goal, but also our brain while we're waiting for the hopefully reward of that decision. And here we have areas involved in the medial prefrontal cortex, that is sort of more the middle section of the frontal lobe, as well as along the side of the parietal lobe as well. So some additional research has been done specific to humans regarding the kinds of more nuanced situations, such as those that are involved when we have to make choices about what we're eating. And these sorts of more complex situations involve changes in value over time. So essentially a shift from short-term value that's very high, but really decreased poor long-term value. And we can think about this when it comes to eating food, right? This is very easy to think about. So when you're really hungry, the short-term value of any kind of food is going to be a lot higher, right? So you're likely to think like, yeah, you know what, like that side of fries, that has a lot of value to me. I'm really hungry right now. And so this is a good decision in the short term, right? But perhaps in the long term, and if you were to continue in those sorts of short-term decisions about fries, for example, that might then lessen the long-term value of that decision because the value is changing over time. It's impacting your body, and that's leading you to evaluate in a different way. So as an example, there's a study that looked at what happened when people who were on diets were given their choice of a tasty but an unhealthy treat, such as a jelly donut, in comparison to a healthy but perhaps less tasty one, like just some plain yogurt. And interestingly enough, the activity in the ventral medial prefrontal cortex, and this is an area that includes the orbital frontal cortex. You can see here, this is a part of the brain that's closest to the eyes in the frontal lobe. The activity in these regions was correlated with taste preference. So this was regardless of whether the item was healthy. So these areas were doing more of a short-term sort of evaluation of, does this food taste good? Yes or no? But in contrast, the area of the dorsolateral prefrontal cortex was associated with the degree of control. And so the activity here was found to be greater on trials in which the person's preferred but not healthy items, so presumably, because of course we have to take into account individual differences, but presumably most people would say that they would prefer a donut over yogurt. So presumably there was higher activation on in the dorsolateral prefrontal cortex when items were preferred but not as healthy. And when somebody had to make a refusal of that combination of foods, there was more activation here compared to when they were choosing the preferred but unhealthy item. And also, interestingly enough, this difference in activity based on refusal and selection was a lot greater in people who were judged to be better at having self-control. And so it's been suggested, taking more of an evolutionary psychology sort of a lens, it's been suggested that maybe the ventromedial prefrontal cortex originally evolved to help us determine the short-term value of something. But then over time, structures like the dorsolateral prefrontal cortex began to help humans with our ability to incorporate long-term considerations into the value assignments that we were giving to stimuli in our environment. And so essentially, we're seeing this pattern of activity that's suggesting that there are different areas involved in the short term versus the long term. So that is that the instant gratification that comes from maybe just choosing whatever meal is available to you in that particular moment because you're so hungry, is a totally different area than is involved with trying to think more longer term and exhibiting more self-control. And so overall, the various kinds of studies that we've been talking about, that is the more neurophysiological and neuroimaging studies, they indicate to us that the orbital frontal cortex plays a really key role in representing value to us. Whereas the more lateral portions of the prefrontal cortex are important for us enacting control on the representations of value. And there's also been a difference between the neurophysiological and the neuroimaging results. So with the neurophysiological approaches to research in this area, that is the kinds of research studies that have used electrodes, that research has helped us get a better picture of how value representation is essentially spread through the different brain networks. whereas neuroimaging has given us a sense of the specialization that comes with the different regions of the brain. So again, the neurophysiology, the electrodes give us a sense of the networks of cells that are involved, whereas the neuroimaging gives us a sense of the different regions and also their different roles in decision making. So obviously, the lab is an artificial environment. Having to bring people in to a scanner is obviously very artificial. And so many of the experimental paradigms that we've been talking about have involved this artificiality. Um, but at the same time, a lot of these studies have also worked to try to give a person an idea of the different choices that they have in that particular situation, as well as at least some information about the potential rewards and costs that are available or that come with a decision. So therefore, participants are in a position where they can calculate and compare values. Um, and even though this is ideal theoretically, this actually isn't the case in the real world. So not only is our sort of lab studies on this artificial in terms of just the sort of setting that you're in, but there's also artificiality in terms of the kind of information that you have available to you. Because often in life, unfortunately, we don't have all of the possible options available to us. And so a classic example that has been used here to try to better approximate the real life situations that often come up, at least in animal studies, has been looking at foraging behaviors. So foraging behaviors, these are exactly the kinds of situations in which um, uh, an organism does not have all of the possible information available to it in that moment to make a decision. And so these animals are having to make decisions about where to seek food and water, if they should enter into maybe higher risk areas that might potentially have a higher reward associated. Um, and so foraging requires decisions such as do I stay here or do I try to move on? So do I continue to exploit the resources that are at hand, or do I set out to explore in the hopes of finding something better? And so here, the animal has to calculate the value of the current option that they have, um, their their perception of the richness of the overall environment, and the possible costs of exploration. And so the result of a number of animal studies has supported the hypothesis that the anterior cingulate cortex plays a critical role in foraging like decisions. And this has been further supported by fMRI studies in humans. And so obviously, even though we are not engaging in the same kinds of foraging behaviors, or at least to the same extent and conditions, such as, being out in a field and trying to consider, well, do I continue walking in the search of trying to find water, or do I try to just stay here? Um, but we still encounter conditions in which we are faced with these kinds of situations where we have to consider what we currently have versus what we might be able to have. The saying, the grass is always greener, kind of comes to mind here of, do I settle for what I currently have, or do I try to see if something else out there is better than what I currently have? And so there have been fMRI studies in which participants have been asked to make choices about where to go in sort of a virtual world. They're put in this sort of VR immersion, and they are asked to make choices about what they would do if, say, they were, I don't know, in a forest or something like that, and they needed to find certain resources. And so in these studies, the activity of the ACC was correlated positively with exploration. So that is, the more exploring that somebody did, the more ACC activity was involved. And so taken together, these sort of animal studies and also these human studies, they suggest that the ACC signals a type of control by promoting or instigating a particular behavior. And that is the choice to explore the environment for better alternatives in comparison to the current course of action. So essentially, we have this brain area that sort of makes us want to push forward oftentimes when making decisions, when we are faced with a situation in which what we currently have might not be as good as what we could possibly have. And so it's not surprising then that the rewards that come from our decision making are fundamental to the behavior of all animals, and obviously humans included in that. And so it makes sense that we might expect the processing of such signals to involve the older neural structures, what is often referred to as the quote unquote reptilian brain. And this is the case. Has been a lot of evidence to indicate that many of the subcortical areas that are considered quote unquote older, that these are the areas that represent reward information. And so this includes the areas of the dorsal and the ventral striatum, the hypothalamus, the amygdala, and the lateral habenula. You don't need to worry about memorizing those, but just wanted to point out that there is a certain group of structures that seems predisposed to considering rewards in the context of decision making. And a lot of the work on reward has focused on the neurotransmitter dopamine. It's important to keep in mind that the reinforcement process likely involves the interplay of a lot of other neurotransmitters as well. But for the purposes of our discussion, we are just going to generally talk about dopamine. And so there are what are called dopaminergic cells, that is dopamine producing cells, that are really scattered throughout the brain. But there are two areas in which these dopaminergic neurons are highly concentrated. So the first area is called the substantia nigra pars compacta. And the substantia nigra is an area of the basal ganglia. And the basal ganglia is a very important structure in the disease process of Parkinson's. And what happens in Parkinson's is that there is a loss of these dopamine cells. So you are in this area that has been highly concentrated with dopamine. And because of the Parkinson's disease process, the concentration of these dopamine cells decreases. And that is what leads to one of the many things that leads to the motor difficulties that are associated with Parkinson's. Anyway, the other area of dopamine concentration is what's called the ventral tegmental area, or the VTA. And the dopamine neurons in the VTA are further organized into two separate pathways. So one of the pathways is called the mesolimbic pathway. And this travels to structures that are really important in our emotional processing. So this includes an area called the nucleus accumbens, the amygdala, the hippocampus, as well as the ACC that we've been talking about. And the VTA also has pathways that travel through or have a pathway called the mesocortical pathway. And this pathway projects to areas of the neocortex, particularly to the medial portions of the frontal lobe. And so, as we've been talking about, we know that these areas that are more frontal in the brain are often associated with more volitional control of our decisions. But also, like we've talked about, they also have to do with the representation of value and how we mentally understand that. So, and just some, this is more just sort of, hey, fun fact of the day. You don't actually need to know this information per se, but just to kind of give you a foundational idea of how dopamine came to be linked to the idea of reward. It has to do with work in the early 1950s by researchers James Old and Peter Milner. And what these researchers did is they implanted electrodes into the brains of rats, and then they gave the rats the opportunity to essentially control the electrodes. So they implanted these electrodes, and if the rat pushed a lever, the electrode would become activated. And so some of the rats, they really rarely pressed the lever. But interestingly enough, there were other rats that just pressed the lever like crazy. And this means that they were just activating those electrodes just over and over and over again. And the difference between these rats turned out to be the location of the electrodes themselves. So the rats who could not stop pressing the lever were the ones whose electrodes were actually activating their dopaminergic pathways. So they were activating some of the exact areas that we've been talking about on this slide. And so these were observations that led to the idea that dopamine was the neural correlate or neural mechanism of reward. And so there have been additional studies that have been done to try to replicate and expand on these findings. And so originally, based on Olds and Milner's research, neuroscientists thought that the stimulation of these dopaminergic areas caused the release of dopamine, and that this event resulted in a pleasurable sensation. And so that was how reward worked. This hypothesis, however, in recent years, it's turned out to be too simplistic. There's then been further research that has been done not only on dopamine, but also on other neurotransmitters that might be interacting with dopamine when it comes to the brain's processing of rewards. So there's also been some other work that has been done to try to distinguish between some of the rules and the mental models approaches that we've talked about behind reasoning. And so we're going to shift briefly to talking about reasoning because, like we've talked about before, reasoning often goes hand in hand with decision making. So, some researchers who have been notable in the area of neuroimaging and reasoning have been Goal and colleagues. And they've conducted studies on adults performing various reasoning tasks. So this has been more things sort of like those more structured categorical syllogisms, what you might think of in terms of very strict logic or philosophy of the if P then Q sort of representations. And they've also looked at what are called three-term series problems. So as an example, Karen is in front of Larry, Larry is in front of Jane, is Karen in front of Jane? And so they had their research participants working on these various reasoning tasks while they were in fMRI or PET scans. So again, these researchers were really trying to look at the brain activity that was going on during these tasks. And these studies found that different areas of the brain are activated for different tasks. And the activation is also dependent on whether the conditions of the reasoning task are more concrete or if they're more abstract. And so the researchers, they interpreted their results as being more in line with more of a mental models kind of approach. And this is because their evidence pointed to significant involvement of areas of the brain having to do with visual and spatial perception. So it seems like, at least in terms of some of the mental models that we were talking about, the normative versus descriptive versus naturalistic, that there are a wide number, there's quite a wide array of brain areas that are involved. And it's drawing on a lot of different areas. It's not like, oh, there's just this one reasoning area, but it seems to be recruiting other areas that are involved in our perceptual abilities. But essentially, overall, just to really summarize a lot of other studies, the neuroimaging research that's been done on reasoning, it suggests that there are at least two different sorts of neural systems that come online when we are engaged in reasoning. And so one of these involves the left hemisphere and comes online when we are consciously and verbally reasoning. So it's thought that these are the areas that come up when you're maybe, say, trying to verbalize through a problem. Maybe you are, whether that's you're talking to yourself while you're trying to solve something, or maybe you're working in a group, you're going to have more of the left hemisphere showing up. And this makes sense, right? Because as a reminder, the majority of people, their language areas are in the left side of their brain. And then for reasoning that is not specifically conscious and verbal reasoning, it seems that both hemispheres are recruited. And so it seems like which system is used, it depends on the kind of reasoning that the person is engaging in, as well as the demands of the task as well. And then there's another study by Waltz and colleagues, and they found that people who had sustained damage to their prefrontal cortex, they really, really struggled in their reasoning abilities when they had to integrate multiple pieces of information. And particularly when these multiple pieces of information were not directly presented in a way that allowed for the person to really easily integrate them. So essentially, this would be the kinds of things where um, if there was like a word problem and it was a lot more complex, people really had a hard time if they had damage to their prefrontal cortex. Um, and Waltz, they showed that there weren't deficits in these people's intelligence, and there also weren't deficits to their semantic memories. So it wasn't that their intelligence or their memory abilities were what was contributing to this, but rather it seems that their prefrontal cortex was no longer able to engage in the process of really gathering and unifying information mentally to then help them reason well. So it would be like if you were trying to problem solve and you usually had a table, right, where you could just lay out the different pieces of whatever you were working on. It would be like if all of a sudden that table was completely gone. Um, and so, again, it seems that there are a lot of different areas of the brain that are involved in reasoning and also our decision making. So I just wanted to end our lecture with a short, interesting video. I came across this. It's from, I think, 2018, so it's relatively newer. And it's an interesting technological development that has been made in the experimental approach to studying decision making.
The brain is like a library where each neuron is an individual book in this library. And the problem we have is that we don't know what the books are saying, and we don't even know what language the books are written in. And without that information, it's going to be very hard, obviously, to figure out how the brain works and what goes wrong during diseases of the brain. I'm Michael Heuser. I'm a professor of neuroscience at University College London. We're trying to understand how neural circuits of the brain process and store information relevant to behavior. So for us, the Holy Grail is to understand the neural code. So 50 years ago, we cracked the genetic code, and what we want to do is try and understand how the pattern of spike firing in neural circuits of the brain represents information and stores information. So neuropixels are a new kind of silicon probe with amazing capability for recording the activity of the brain. So these are silicon probes, which are thinner than a human hair and have 1,000 recording sites and allow you to record from more neurons than ever before. Because the traditional way of recording brain activity is to record it one neuron at a time. So this approach is a major breakthrough because we can record from more neurons than we can ever record from before. So it's going to give us unprecedented information about how the brain works, especially in decision making. The structure and function of the neural circuits of the mouse brain appear to be very similar to those in the human brain. Moreover, mice are actually pretty smart. They can make quite sophisticated decisions, and we can use them for quite cognitively challenging decision-making tasks. The mouse is navigating in a virtual reality environment to try and reach a target. So the real world equivalent of that is, for example, navigating through the streets of London to reach a material board. This requires integrating visual cues with knowledge of the environment and a cognitive map which represents the world inside the circuits of your head. We're also now able to explore how different brain areas are interacting together and sharing information between each other. However, even though this is a dramatic leap, we're still only recording from a small fraction of the neurons that are active during decision making in the brain, even with this remarkable new technology. And so the challenge is to further scale up this technology and also develop other new technologies. To fully understand diseases of the brain like Alzheimer's, schizophrenia, autism, we first need to understand how the neural circuits of the brain work. And once we have that information, this will give us a handle on what's going wrong in these diseases.
So that's where we're going to stop for today. We'll continue talking about some of the more real world implications of decision making when we meet for class on Wednesday. See you then.